Good morning, everybody. My name is Mateusz Jundzill. I'm a PhD student in Universitat Klinikum Jena in Germany, and I would like to present my poster, Comparison of Prokaryotic Annotation Tools using 14,000 species. Are you getting the most out of your genome? The aim of this project was to investigating the performance of 4 different bacteria annotation tools that are commonly used: BACTA, PJOP, Proka, and Eggnog Mapper. And for this tool, I will use the shorter name Eggnog in the future. We compare the performance via 3 different characteristics. The general tool performance via checking the composition of annotated space, the functional annotation that we check via GO term count, and the genome quality by checking the stability of the annotation after introducing different levels of deletions to the provided sequence that we call noise. The general tool performance we investigated via the checking the annotated coding space. And all of the tools basically perform similarly in case of predicting the full and hypothetical annotated features on comparable amount of space. Here denoted as a dark blue and the light blue. However, there was some divergent observation, especially in the Venericutus filium, that I will talk about it in the later. A little bit later. Overall, BACTA has the lowest proportion of coding length that was occupied by the hypothetical proteins, the light blue one, and that basically does not provide, only knows that the gene exists up there, but it does not provide any information about what this gene actually is, which is actually especially evident if we compare the results from between BACTA and Proka, where in case of Proka, almost half of the coding space is occupied by the hypothetical genes. We also checked the count for RNA and tRNA, and all of the tools basically perform similarly, except Eggnog Mapper that does not provide the function, this particular function, so it was excluded for analysis. And as we look a little bit closer in the class, in the annotation composition between different classes, and we focus about the Stenericutus philia that I was talking before, we observe major differences in coding space that was annotated between PJOP that performs on par with the other classes, and BACTA, Proka, and Eggnog that perform much worse in comparison to the other classes. And the same results are replicated if we focus about the hypothetical genes. Here again, PJOP performs the best, and BACTA and Eggnog perform a little bit worse in comparison to PJOP. Moving forward to the GO term annotation, basically Proka and BACTA did not annotate anything meaningful here. So we will focus only about the results from Eggnog Mapper and PJOP. And PJOP have the most amount of genes that have at least one GO term. Yet, if we actually focus about how many GO terms we have per gene, then Eggnog is clear winner, and we have the actually the median GO terms per gene that could be meaningful for any downstream analysis, because the GO term count of 2 basically does not provide anything meaningful for the downstream analysis. In case of the stability of the annotation with different level of deletions, we investigated 3 different levels: 0.5, 1%, and 2% deletions of the original sequence. And we observed that PJOP and Eggnog Mapper demonstrated the highest levels of highest stability under increasing the noise. And to our surprise, BACTA is really sensitive to any noise, and even on the smallest level of the introduced noise, so this 0.5%, we observe really high spike of the proportion of hypothetical proteins, and the results are basically comparable to the results of Proka. What is also a little bit interesting, in case of PJOP, even on all of the levels, we observe an increase of the coding space that was annotated in any noise level in comparison to the original sequence in some particular cases. We will investigate it in the future, actually why it's happening and what's going on there. But it's also something that we didn't observe in any other tool cases. And to summarize my findings, basically Bacta performs superior due to minimizing hypothetical genes in case of the good genome quality, and if we exclude some particular taxonomical cases. EGNOG mapper performed the best in the functional gene annotation, and PJOB displays superior annotation stability if the sequence quality is low or is performed on some specific taxonomical classes. The further analysis of the factors that could influence the tool performance are currently investigated and will be processed for publication in the future. Thank you for listening. You can find me on ISMB ACCB 2023 conference in Lyon. And of course, I'm also reachable for any questions via email or ISMB platform. Once more, thank you for your attention.